Okay, hello everyone. Welcome to BIOL 66 Final Exam Review Workshop. My name is Jason. I am one of the peer tutors for the CHHS Student Success Center.
And my name is Stacy Mertzis. I am also a tutor for the center.
So a little bit about this workshop. In this workshop, we're going to cover some important concepts from chapters 17, 18, 19, and 20. And we picked out these specific chapters because we've hosted workshops previously for the other chapters, right? All the chapters before this workshop. So in chapter 17, we're going to cover renal physiology, like the renal functions, urinary pathway. And chapter 18 for the digestive system, we're going to talk about, again, some of the general functions and the functions of the GI tract structures, like what's the difference between the alimentary canal and the accessory organs. And I'm going to dive into the digestion of the three macronutrients, right? The carbohydrates, proteins, and lipids. And in chapter 19, for regulation of metabolism, we're going to go over absorptive and the post-absorptive states, and also talk about diabetes mellitus. In chapter 20, reproductive system, we're going to cover sexual reproduction and a little bit about reproductive endocrinology. And keep in mind that your final exam is this Friday, December 10th from 12:15 to 2:30 p.m. All right. So throughout this workshop, we're going to have some polling questions, and this is just to have you allow you guys to answer the practice questions and check for learning or check for understanding questions through the poll feature. So in this practice question, I'm going to launch a poll, and you guys can have a chance to answer this. Are you guys able to see the polls? Okay. I'm gonna give you guys about like 30 seconds for this question. And yes, as Stacy mentioned, we do have a survey for you to complete. And if you fill out the survey at the end of the workshop, we will email you a copy of the workshop slides. And the previous workshops that we hosted for the other chapters are already posted on our YouTube channel. And the link is attached on the chat right now. Okay, so I'm gonna end the poll in like 10 seconds. Okay. That was pretty easy, right? Right. So your final exam is on Friday, December 10th. Let's jump into renal physiology.
Yes. All right. Thank you, Jason. So we're going to do kind of a high-level overview of renal anatomy and physiology, particularly the renal functions, and then we'll go over the general urinary pathway. Next slide. All right. So renal, when we say the word renal, I want you to think of kidneys because that's what renal means. And one of the general functions of the renal system is to regulate extracellular fluid. There's also many other functions such as like regulation of blood volume, electrolytes, pH balance. We don't have time to kind of go super deep into that, so I'd refer you to your lecture slides, but just kind of one of the general functions that you should know is that it regulates the extracellular fluid. So as far as the anatomy and physiology behind the renal system, there are typically two kidneys within the human body. And so these two kidneys work to form something of the blood. What do these two kidneys do? Oh, and you guys already know. Yeah, put your answers in the chat. Feel free to unmute and share your thoughts as well. And then what I'm going to do is I'm going to kind of type up the answers that you guys give me so we can all see them on the screen. And then after this workshop, I'll go ahead and input the answers so that for those of you that fill out the survey, you'll be able to see the fill in the blanks. All right, I see some comments in the chat saying filtrate, absolutely. So the two kidneys work to form a filtrate of the blood. And so with this filtrate of the blood, some of that blood is going to be blank, and then some of it is something else is gonna occur. What are the two things that the filtrate? So I see a comment in chat saying reabsorb. Yeah, absolutely. And then other comments saying secreted and reabsorbed. Absolutely. You guys are absolutely correct. So some is secreted, and then other blood is reabsorbed. So secretion and reabsorption, as far as the function of like where that filtrate is going, we're gonna go more into detail on that in a moment. But this is just kind of the overview that the two kidneys form a filtrate of the blood. Some of that filtrate is either continues to be secreted through the renal system, or it is reabsorbed back into the blood vessels. So when we think about the filtrate that is secreted and it continues through the renal path, eventually we're going to be forming urine, right? And so urine that is formed, that is destined for what exits the kidneys? What is the word that we're looking for? I see a lot of comments in the chat saying excretion. Absolutely. So excretion. So a lot of students, and I know when I took the class, I would always get confused between secretion and excretion. Basically, when you hear secretion, think about, the blood filtrate. And so where is this blood filtrate going? Is it being secreted into, and we'll look at later the specific structures, but secreted through the renal system? And then versus excretion, think about excretion in forms of exiting, right? So this urine, this waste products that are formed within our renal system, we want them to leave our body. And then by definition, excretion is to leave. So excellent job answering those correctly in the chat. All right, so let's think about it in terms of blood, because we said the two kidneys form filtrate from the blood. So blood is going to enter the kidneys via the renal, which vessels? I'm seeing in the chat, arteries, absolutely. And think back to when you guys learned about the cardiovascular system, right? Like arteries are vessels that carry blood away from the heart, right? And so like renal arteries, like we're not going to the heart, right? We're exiting the heart and we're going to the kidneys, right? And then blood that exits the kidney is going to exit that kidney via I forgot to put the word renal, but what is the other vessel? I'm seeing in the chat renal vein. Absolutely. You guys are absolutely correct. Good job on that. All right, so this question was pertaining to blood. So just to reiterate, blood is going to enter the kidneys via the renal, which vessel was that again?
The renal vein.
Close. It's the opposite. It's entering the kidneys.
Renal artery.
Yes, absolutely. So yeah, so similar, it's matching the question above. So blood is going to enter the kidneys via the renal artery. And then in the one above, we talked about, okay, blood is going to enter the kidney via the renal artery, and it's going to exit the kidney via the renal vein. But in this question below, we're not really focusing, we answered the first part, right? That the blood is going to the kidneys via the renal artery. But read this question carefully. Then we're going to switch and talk about urine, right? The urine that was formed within the renal system. Then the urine is going to exit the kidney via the, see a comment in the chat saying the ureters. So that answer is correct. I feel like there could also be a more correct answer. If you look at this image here, urine is going to exit the kidney like via the renal pelvis and then the ureter. So the ureter is not an incorrect answer. Renal pelvis might be like the more correct because it's like directly before the ureter. So with that, I would recommend going in your lecture slides and maybe seeing like which terminology Dr. Anand uses and like which specific structures that are really important for you guys to learn. But yeah, ureter is not incorrect. I just wanted to point out that in this image here, the renal pelvis comes directly before the ureter. All right, so I'm gonna go ahead and clear the annotations. And again, I'll add these for those of you that fill out the survey, you'll get the fill in the blanks as well. All right, next slide. All right, so let's dive into how the kidneys function, right? So the previous slide we just worked on was kind of an overview of the general functions of the renal system, how they're gonna filtrate the blood, some of that's gonna be secreted, some's gonna be reabsorbed, and then eventually the urine that's formed, like where's that gonna go, right? It's gonna exit the kidney and eventually get to the ureters. So let's kind of think deep within the kidney. How is this going on, right? So the functional unit of the kidney, can somebody tell me the name of that?
The nephron.
Yeah, absolutely. Good job. So the nephron. And there are, oh, why didn't that text pop up? Okay, so there are tons of nephrons in the kidney and they all work to help the renal system function. So there are two main components of the nephron. And right here in this image, I have an image of like an uncoiled nephron and how some of the vessels, like the arteries and the veins that we talked about in the previous slide, come in contact and connect with the nephron. So there are two main components of the nephron. Can somebody tell me what those two components are?
The renal corpuscle and the tubule?
Yeah, absolutely. So we have the renal corpuscle and the tubules. Can somebody tell me what the renal corpuscle is? What are there's two, so there are two main components of the nephron. You guys answered those correctly, but then let's really quickly focus on the renal corpuscle. What are those two components? Start with a G. Yeah, absolutely. So the glomerulus, which I'm going to annotate. The glomerulus is, I'm highlighting it in pink. And then the capsule is around it. Oh God, these are bad highlights. I'm sorry. But yeah, the glomerulus and then the Bowman's capsule, otherwise known as the glomerular capsule. So that is what the renal corpuscle is. All right, we'll continue with the question. So the glomerulus, right? The glomerulus is a network of what within the glomerular capsule? Oh, thank you, Jason.
Capillaries.
Yeah, absolutely. And this makes sense because when we think about renal physiology, it involves blood, right? We're filtering the blood. And so if the nephron is the functional unit of renal physiology, it would make sense that, you know, capillaries are very much involved because it's dealing with blood. So the blood that enters the nephron, the blood that enters this functional unit, 20% of that blood is going to become filtrate and eventually be secreted and then forms, you know, urine, which is excreted. So it's like really the heart of, you know, the purpose of the renal system, or at least one of the purposes of the renal system. So 20% of that filtrate, 20% becomes filtrate. 80% of that blood is blank back into the vessels. So I'm telling you that 80% of blood is going back into the vessels, but what's that special vocab word that we think about when we think of like back into the blood vessels? So seeing in the chat, reabsorbed, absolutely. So reabsorption, think into the blood. And so right now we're talking about renal physiology and how, blood filtrate is either secreted or reabsorbed. But then also what comes to my mind, and you guys might remember, is that think about the digestive system, right? When we eat food and it gets digested in our GI tract, we need those nutrients, right, to live and have our bodies function. And so those nutrients are being, absorbed, right? And so when food breaks down, and Jason's going to talk all about this later, we're going to absorb those nutrients. And when we think of absorbed, it's meaning like to the bloodstream. So yeah, reabsorb. Excellent. So filtration is the movement of solutes from the glomerulus to the blank and then the blank. So what are the two structures that the filtrate is going to hit right after the glomerulus? Because the glomerulus is where filtration occurs, right? Glomerular filtration. But then where's that filtrate going to go?
I think the first one is glomerular capsule.
Yeah, absolutely. So capsule. And then right after the glomerular capsule, what's the next structure? Think of, I guess it's more of an anatomy question. Like think of the pathway.
The proximal convoluted tubule.
Yeah, absolutely. And I think for general too, like I would have even accepted tubules, but I like how you got very specific.
And is the proximal convoluted tubule of the nephron?
Yes, it's part of the nephron.
You.
Good question. All right, perfect. So thank you guys for answering those. I'm going to go ahead and clear those annotations. All right, Jason, you can go to the next slide. All right, so really quickly, let's kind of run through the pathway of blood and then eventually filtrate through the renal system. So we already talked about how arterial blood is going to enter the kidney through the renal artery. And then arteries, these arteries, right, these renal arteries are going to branch to form, and this is a multiple choice question. So whatever I have in bold, you have to pick one of them because only one of them is correct. And I'll annotate on the screen. So arteries branch to form blank arterioles. And at this point in the pathway, is it the afferent or the efferent? Think about the definitions of those two. I'll help you guys out with this one. So the arteries branch to form afferent arterioles. And your clue is that like, right, A arriving, we are, afferent. So the arteries branching in this image here, right, the arteries are going to connect with the afferent arteriole. And this arteriole is going to deliver this blood in step 3. The blood is going to enter the glomerulus. So the afferent arteriole delivers that blood to the glomerulus. As we talked about last slide, 20% of that blood is going to filter out of the glomerulus into the what? Your keyword here is the filtration, right? The 20%. We talked about how 20% is secreted. Where is that going? Is it going to the capsule or is it going to the peritubular capillaries?
The capsule?
Yeah, absolutely. And we'll see later why it's not the peritubular capillaries. That kind of has a different function. So yeah, good work. So it goes to the glomerulus to the capsule. And so when we think about the definition of glomerular filtration, it's that, right? This secretion from the capillary bed of the glomerulus into the capsule. So now that we're in the glomerular capsule, this filtrate is going to travel to the tubules, but which one? Does it go to the proximal one first, or does it go to the distal one first?
Proximal.
Yeah, absolutely. And your key here too is for those of you that have taken anatomy, by definition, proximal means closer to the point of attachment. And so if we're focusing on the glomerulus, well, the proximal convoluted tubule is the one that's close to the glomerulus, right? And the distal one is the one that's a little bit farther. And this image shows that here. So after it travels through the proximal convoluted tubule, it's then gonna travel through the loop of Henle right here. And then it's gonna hit another tubule. And what do we call that tubule?
Distal.
Yes. So the distal tubule. And then after all this secretion and urine formation is happening within the nephron, it's going to go through the collecting duct, and then it's going to hit the renal pelvis. And then this urine is going to travel to two main structures. And I have this tiny little image here to guide you. After it's exiting the kidneys, what are the two structures that this urine is going to travel through and essentially be stored in before micturition? And by definition, micturition means to urinate. So what are those two structures? Feel free to unmute or put it in the chat. See in the chat the ureter and the bladder. Absolutely. So the ureter are these tubes that transport the urine from the kidneys to the bladder. And so I always like to emphasize, when I took anatomy, they always told us, emphasize that it's the urinary bladder, because there's other bladders like the gallbladder. And so even though it's like kind of obvious what we're talking about, they just like engraved that in my head to like always specify. So yeah, ureter and the urinary bladder. Absolutely. All right, so eventually for micturition, AKA urination to occur, or the expulsion of urine from the body. All right, so that's the kind of the secretion portion of it. So blood remaining in the glomerulus, and we've talked about how that's about 80%, it's gonna leave the nephron. And you know how up here we talked about for step 2, how the arteries form afferent arterioles and the blood is gonna enter the glomerulus? Well, like we said, 80% of that blood doesn't want to keep going like through, the nephron and, doesn't want to form urine and be excreted. It wants to be, reabsorbed into the bloodstream. And so which arteriole is going to be responsible for that at this point?
Efferent arteriole.
Yeah, absolutely. And then your clue is, oh, not that, I need to draw it. Your clue is E exiting, right? We're exiting the nephron. And so they're going to be reabsorbed into the what? Are they being reabsorbed into the capsule or reabsorbed into the peritubular capillaries, which are colored here in orange in this image that I drew? Which structures is responsible for reabsorbing the blood? See in the chat, capillaries. Absolutely. Yeah. So specifically the peritubular capillaries are responsible for reabsorbing that blood that doesn't, secrete. And that's about 80%. And then it's going to exit the kidney, right? These capillaries are going to pick up that blood. And then think about when you learn about cardiovascular system, like the transition between like arteries to capillaries to veins. So if the peritubular capillaries are reabsorbing that blood, and we're exiting the kidney, on previous slides, we talked about how the renal veins, right, are the specific vessels that are going to deliver that blood away from the kidney. All right, excellent. Good work answering those in the chat. And thank you for your verbal answers as well. I really appreciate it. All right. All right, next slide, Jason.
Okay, so now I'm going to go into the digestive system. And in this chapter, I'm going to briefly cover some of the general functions, some structures, and the digestion of the 3 macronutrients. So obviously, we know that the fundamental function of the digestive system is to break food down, right? It's the breaking food down into molecular forms that are then transferred or absorbed into the internal environment, also known as the bloodstream. All right. So first we have motility. Motility sounds very much like mobility, right? So motility in the digestive system refers to the movement of blood or movement of food through the gastrointestinal tract. Okay. And then obviously that digestion is the chemical breakdown of food from macromolecules into smaller or micromolecules so that it can be absorbed. And we also have secretion. So secretion literally means the release of certain fluid or certain hormones into the GI tract, right? So secretion means the release of endocrine or exocrine hormones into the lumen of the gastrointestinal tract to aid with digestion, to help with digestion. Now, my question is, what is a lumen? Does anybody remember what a lumen refers to? What is meant by the lumen of the esophagus or the lumen of the small intestine? Does anybody remember? Okay, so when we talk about lumen, right, think about the channel or the opening within a tube, right? So let's say the esophagus. When we talk about the lumen of the esophagus, think about the channel inside of it, right? That opening, that channel, that is being held within the epithelial cells, like right, the linings of the esophagus, right? So, and when we meant secretion of endocrine, exocrine hormones, those fluid or hormones, they're released into the tube, right? Into the channel of the tube. So that's what we meant by lumen, right? It's just literally just like an opening. Moving forward, we have absorption. Right? Absorption refers to the transport or the movement of digestive products into the internal environment, into the blood. And then our digestive system can also serve as storage and elimination, right? For example, the stomach can temporarily store food, and the system can also eliminate indigestible food molecules, right? So you can get rid of food that cannot be broken down. And it can also act as immune barriers, right? Because of the tight lining of the small and large intestines. So, you know, it can act as a physical barrier to toxins, right? So these are just like some basic terminologies and functions of the digestive system, okay? Okay, so now I'm moving on to the gastrointestinal tract or the alimentary canal. This is more of a anatomy review, so I'm going to quickly go over this. So what organs and tissues make up the GI tract and what makes it unique? So the GI tract or the alimentary canal consists of the mouth, the pharynx, the esophagus, stomach, small intestine, and large intestine. Okay, and it's called the alimentary canal because it's this whole tract that starts from the mouth through the pharynx, through the esophagus, into the stomach, into the small intestines, and then goes to large intestine, and then out through the anus. So it's this whole 30 feet long sort of tube that's the alimentary canal, right? Now, can anybody tell me where does most of the digestion and absorption occur? Does anybody remember where most digestion and absorption takes place?
The small intestine.
Exactly, right. So small intestine is where most of our digestion and absorption occurs, right? I'm going to talk more about this in later slides. So this is just the gastrointestinal tract or the alimentary canal. Now, how does it differ from the accessory organs? Well, the accessory organs are the teeth, the tongue, the salivary glands, the liver, the gallbladder, and the pancreas, right? The teeth, the salivary gland, the tongue, the liver, the gallbladder, and the pancreas, right? These are the accessory organs, right? And these accessory organs, they help with digestion, right? They secrete substances into the GI tract to help with digestion. And keep in mind that they are not parts of the GI tract, but are rather organs or tissues that play a role in the digestive process. Okay. Another question. So does anybody remember what is the yellow-green fluid that is produced and released by the liver and is stored in the gallbladder? What is that yellow-greenish fluid called? Does anybody remember? It's produced by the liver and released by the liver, but is stored in the gallbladder.
Bile.
Bile, exactly, right. So the bile fluid is produced and released by the liver and is stored in the gallbladder. Now, what is the main function of this bile fluid? Does anybody remember? Like, what does it break down? What does it help to? What does it help to digest?
Is it protein?
Not proteins. It's another macronutrient. Right, we have three macronutrients, right? We have carbohydrates, lipids, and proteins. And bile is secreted to help digest one of those macronutrients. Esther said fats.
Yeah. Sorry, yeah, fat.
Yeah, exactly. So bile is secreted to help break down fats into fatty acids or into smaller fat globules, right? So that it can be digested by lipase enzymes. Okay, so that's, and I'm gonna go in detail of that when I talk about the digestion of lipids. Okay, let me clear my drawings. Now I'm gonna launch a poll, and this is just like a check for understanding question. So the gastrointestinal tract includes all of the following except what? And I will give you guys 30 to 40 seconds for this. Okay, 10 more seconds. All right, end the poll. Okay, so the correct answer to this question is B, liver. Keep in mind that the gastrointestinal tract starts with the mouth, right? Starts with the mouth to the pharynx, to the esophagus, and into the stomach, and into the small intestine. And liver is an accessory organ, right? It produces bile and releases it so that it can help with the digestive process. So which is why, and keep in mind that accessory organs are not part of the GI tract, but they're just organs that help with the digestion, right? They secrete substances to help with digestion. Okay. Next question. True or false? The stomach is where the majority of digestion and absorption occurs. Okay, I'm gonna give you guys 10 more seconds. Okay, I'm gonna end the poll now. So the correct answer is false, right? I talked about how the small intestine is where the majority of digestion and absorption occurs, right? Well, yes, there are some sort of digestion taking place in the stomach, but a majority of the food breakdown process is in the small intestine, okay? So now let's dive into the digestion of carbohydrates. So carbohydrates is one of the most common and important nutrients that we consume on a daily basis, right? When I think about carbohydrates, I think about rice, sugar, candy, right? Things like that. And so when we consume carbohydrates, it is mostly consumed as starch, okay? Starch is a polysaccharide of glucose. Now, poly means many or multiple. So when you come across the word polysaccharide, think about like these long chains, these multiple chains of glucose, right? And this is where carbohydrates first enter our body when it's first consumed. Now, there is an enzyme that is responsible for the digestion of carbohydrates. Does anybody remember what that enzyme is called?
Amylase.
Amylase, exactly. So amylase is an important enzyme responsible for digestion of carb. So first, salivary amylase. So salivary amylase, obviously in its name, saliva, is where the digestion of carbs begins. Okay, so salivary amylase is an enzyme that begins the digestion of carbohydrates. And it starts from the mouth all the way to the upper stomach, or you can say from the mouth to the end of the esophagus tube, right? So this is where digestion of carbs starts. Next, we have pancreatic amylase, okay? Pancreatic amylase are released into the small intestine, into the lumen of the small intestine. And small intestine is where most digestion of carbs occur, right? Now, in this phase, these polysaccharides, remember I talked about the long chains of glucose that we intake as. So these long chains of glucose, these polysaccharides are broken down into smaller and shorter chains of glucose. Okay, so poly means many, multiple long chains, right? And they're going to be broken down into disaccharides or trisaccharides, right? Di means two or double, and tri means three, right? So these long chains of glucose are broken down into shorter chains, into disaccharides and trisaccharides and etc. Okay, and this is the function of the pancreatic amylase. Okay. Yes, pancreatic amylase is secreted into the small intestine. So next we have brush border amylases. Brush border is part of the small intestine, right? So these these are in the small intestine, okay? So these brush border and amylases are also part of the small intestine, where these smaller chains of glucose, right? Remember we talked about the disaccharides and trisaccharides. These shorter chains of glucose are hydrolyzed into their monosaccharide components. Okay, and remember, mono means single, right? So these single chains of glucose, right? Now, when I said hydrolyzed, so it means hydrolysis, right? Does anybody remember what hydrolysis mean? What is meant by hydrolysis, or to hydrolyze something? A hint I would give you guys is it has something to do with water, Exactly. Yes. Thank you, Esther. So to hydrolyze something, you're breaking a substance down. You're breaking something down due to the reaction with water. Okay. So the purpose of these brush order amylases is to break these shorter chains of glucose, these disaccharides, into monosaccharides. Okay. Now, if this is confusing to you, just keep in mind of the key idea here. Okay. Here's the key idea. The purpose of carbohydrate digestion is to break it down into their monosaccharides component so that it can be absorbed in the bloodstream. Okay, right? They're first consumed as polysaccharides, these long chains of glucose, and they're broken down into disaccharides or trisaccharides, and then it goes to even shorter chain, which is these single chains of monosaccharides. Okay, so just keep that in mind. Is there any questions about this? Okay, let's move along. This is a little flow chart that I pulled up, right? So first, carbohydrates first consumed as starch, and salivary amylase begins that digestion in the mouth and to the end of the esophagus. To begin the digestion of carbs, right? And then pancreatic amylase is secreted in the small intestines to break those polysaccharides down into their disaccharides form. Okay. And then lastly, they're broken down into their monosaccharides form by the brush border amylase. All right. Okay. Now, let's talk about the digestion of protein. So protein is another macronutrients that we consume, right? When I think about protein, I think about chicken, beef, eggs, etc. Okay, so unlike carbohydrates, the digestion of protein actually starts in the stomach rather than the mouth. Okay, so that digestion of protein starts in the stomach. Now, pepsin is an important enzyme for when we talk about protein here. Okay, so pepsin is an enzyme that breaks down the protein into short chain polypeptides. Okay, so pepsin is important. It's an enzyme in the stomach that produces or breaks down this protein into their short chain polypeptides. And again, poly means many or multiple, right? So where you see polypeptides, think about these chains, these multiple chains of amino acids that are bound together. Okay, remember, amino acids is what makes up polypeptides and proteins, right? Amino acids, the bases. Okay, and polypeptides is a molecular form in which the digestion of protein begins. Okay, now in the small intestine, the pancreas releases many different enzymes to further break down these polypeptides so that it can be in even shorter chains to continue the breakdown process, right? These enzymes include trypsin, chymotrypsin, elastase, carboxypeptidase, and aminopeptidase. So I'm not too sure if you need to memorize all these enzymes, but what I was trying to understand here is the breakdown process and right from polypeptides to amino acids and how it's absorbed, right? I wouldn't put too much effort into memorize the enzymes, but rather how the breakdown processes work. Okay. So of course, again, these pancreatic juice enzymes break down the polypeptides into amino acids, right? We want to keep breaking it down to smaller molecules. And remember, polypeptides are essentially these chains of amino acids bound together, right? And these pancreatic enzymes help to separate those amino acids. Okay. So these amino acids that are broken down are then actively transported into the epithelial cells of the small intestine. Okay, so once the amino acids are separated from the polypeptides, they're actively transported into the epithelial cells of the small intestine, and then they're secreted into the interstitial fluid and absorbed into our capillaries. All right. And if this is confusing to you, I will always try to remember the key idea here. All right. So the key here is, so the digestion of protein involves the breakdown of polypeptides into amino acids, which would be then absorbed into the bloodstream. All right. And it's just this breakdown process that just keep repeating itself, right? Breaking polypeptides into even smaller, into their micronutrient form, into micromolecule, into their amino acids, right? Breaking the chains apart. Okay, does that make sense? All right. Any questions about this? All right, moving along. So the third and final macronutrient that I will go over is lipids, or we call it fats, right? So digestion of lipids occurs in the small intestine. Okay, so the digestion of lipids occur in the small intestine. So the presence of lipids in the small intestines stimulate the bile fluid, right? We talked about this in the previous slides. Small intestine stimulates bile, stimulates secretion of bile, which is a fluid that helps emulsify fats, right? Now, what is meant by the term emulsify? Like, what is meant by the term emulsification? Does anybody remember? Okay, I see one response so far. I see two responses. Okay, so emulsification, or to emulsify fat, right? You're pretty much breaking it down from larger lipid molecules into smaller lipid molecules. You're not necessarily thickening it, but you're breaking it down so it increases the surface area so that it can be better digested, right? So the bile fluid is secreted due to the presence of lipids, and bile helps to break down fat, to break it into smaller triglycerides. And this allows for greater surface area for digestion. Now, once the bile emulsifies the fat, the two main enzymes here that helps with the digestion of lipids are lipase and colipase. Okay. Right. So pancreatic lipase and colipase are the primary enzymes here that helps with digestion of lipids. Now, as a result, these enzymes liberate free fatty acids and monoglycerides. Okay, so lipids and colipase break down this triglycerides into free fatty acids and monoglycerides, right? And remember, mono means single, these single chains of lipids, right? So fatty acids and monoglycerides. Once they're broken down, they enter the epithelial cells of the small intestine. Okay, they enter the epithelial cells of the small intestine, and they're combined together to form triglycerides. So the triglycerides in this case, right, we're talking about where we're inside the epithelial cells of the small intestine now, right? The triglycerides in this case is not what we talked about in the small intestine, or no, sorry, not in the, when we talk about when we're first into the small intestine, but we're talking about when we're talking about the inside the epithelial cells of the small intestine. Triglycerides is the combination of the free fatty acids and monoglycerides. Okay, they're combined together to form triglycerides. Now, when they're combined to form triglycerides, that triglycerides combine with protein and cholesterol to form chylomicrons. Okay, and chylomicrons is the only form of lipids or fats that can enter lymphatic vessels and veins. So it is only in the form of chylomicrons that fat can enter our internal environment. Okay, not fatty acids, not monoglycerides, not triglycerides, right? But chylomicrons. Okay. And the next slide, I have a better visual for this. So again, triglycerides enters our small intestine, the lumen of our small intestine, and bile salts detects the presence of triglycerides, stimulates the release of bile salts, these bile fluid, and these bile salts emulsifies the triglycerides into their smaller components, right? And with lipase and colipase, they're broken down into fatty acids and monoglycerides. Okay. Now, fatty acids and monoglycerides then enter the epithelial cells of the small intestines. We're no longer in the lumen, right? This is a lumen, but we're inside the epithelial cells now. We're inside the cells, fatty acids and monoglycerides combine together to form triglycerides. Okay. And this form of triglycerides combine with protein and cholesterol to form chylomicrons. All right, and chylomicrons is the only way in which fats can enter our internal environment, internal system. Does that make sense? Do you have any questions about this?
What are triglycerides again?
Triglycerides, tri means like 3, right? So it's like these basic, like it's like a form of lipids that it's, how do I explain triglycerides? Because I know triglycerides is like the macro form of fat, and then fatty acids and monoglycerides like smaller and like micro molecules of
I think
fat.
it's like kind of complicated into like the chemistry of it. 'Cause from I remember from nutrition, I think it depends on the number of fatty acids, but I don't particularly remember.
Yeah, I would say triglycerides. So they're a type of fat, so a type of lipid in your body. Typically, they're associated with like the stored fat. There's a slide in your lecture. I forget what number it is, but we're gonna talk about metabolism very soon, and in chapter, I don't know what chapter number it is, but the metabolism chapter, there's like a chart that shows like triglycerides break down into fatty acids. And so basically triglycerides are like, they're like fatty acids are like really simple and triglycerides are really complex.
It's like triglycerides, like the macro form, the big form of fatty acids, and they're made up of these fatty acids and monoglycerides. Right. Does that make sense? Does that help?
Yeah. Thank you.
Okay, awesome. Now, another check for understanding question. What is the molecular form in which carbohydrates are typically absorbed into the bloodstream? And I give you guys minutes for this. Yes, 15 more seconds. Okay, I'm going to end the poll now. So majority answered D and some answered B and C. So the correct answer here is D, monosaccharides. Remember the key idea that I mentioned earlier. When it comes to the digestion of carbohydrates, think about, these long chains of glucose, these polysaccharides. They're broken down. They must be broken down into like more simple form, into these single chains of glucose, right? Or else it can't be absorbed into the bloodstream. And monosaccharide, mono means single. So monosaccharide means single chain of these glucose, and they're the most simple form, right? So it can't be disaccharide because disaccharide it's more complex, right? They have two chains rather than one chain. It's not as simple as a monosaccharide. So the correct answer to this question is D. Now, next poll, fats are secreted via exocytosis from the epithelial cells of the small intestine in molecules called what? Or to put it another way, what is the only form of fat molecules that can enter the internal environment? And the key word here is they're secreted via exocytosis. So they're leaving the epithelial cells. They're not entering, but they're leaving the epithelial cells. Okay. You guys can have about 10 more seconds. Okay, I'm going to end the poll now. So I see some various answers. I see A, B, C, and D. Okay, so I'm going to go over this real quick. So the correct answer is C. Remember, chylomicrons, let me go back a few slides. Remember, chylomicrons is the only form of fat molecules that can exit the epithelial cells of the small intestine, right? Fatty acids are what enter the epithelial cells of the small intestine. So the answer to the question must be C, chylomicrons. Okay. And I didn't mention dipeptides or low density lipoproteins, so those answers wouldn't be correct. All right. Next slide. There we go. Metabolism.
All right, so we're going to do a brief overview of metabolism. Basically, we're going to define it and then talk about how it is regulated within our bodies. Next slide. All right, so the definition of metabolism, it's that it is all the chemical reactions that occur in the body. And so that's a very broad definition. But a tip that I like to remember is conceptually, the purpose of metabolism is to provide energy to our tissues and our organs. Our cells, tissues, organs, they need energy to function and help our body survive. So in metabolism, there are two functional states. There are two kind of ways that energy is provided to our tissues. And they're providing the energy for cellular activities, right? The cells are going to take in that energy, and they're also going to regulate blood glucose. And we'll get into the details of that very soon. So the two main functional states are the absorptive state, otherwise known as feasting, and the post-absorptive state, otherwise known as fasting. And so what I want you all to answer for me is, let's first focus on absorptive state, feasting, right? So feasting is referring to like we are consuming a meal. So what function is associated with each state? So a hint to that is there's two answers for this. Think about the absorptive state, otherwise known as feasting, right? And this refers to a functional state of metabolism, otherwise known as, providing, we're getting energy some way, somehow to then deliver to our cells, tissues, and organs. So how does the absorptive state function? Like what's the details of the energy behind that? So you can put the letters in the chat or you can speak up, but my hint to you all is that there's two answers for absorptive state and two for post-absorptive, but we'll get to that in a minute. So what are the two that are associated with absorptive? All right, I'm gonna annotate. I see in the chat, A is one of them. What else? So I'll tell you A is correct because when we think about it, absorptive, AKA feasting, if we're eating a meal, right? That meal is going to go through our GI tract, all the digestive processes that Jason covered, that's going to occur. And then as I talked about kind of a little bit when we're talking about what reabsorption entails. So the nutrients that are digested, the nutrients want to be absorbed into the bloodstream. And so when the bloodstream receives those nutrients, the bloodstream, think about the cardiovascular system. It's a transport pathway, right? So if the blood receives nutrients, it's going to deliver those nutrients to our cells and tissues. And those nutrients are, used for energy. So A is correct. Also feasting, I see in the chat, C, anabolism. Yeah, absolutely. So as we're eating, right, anabolism by definition is the creation of energy reserves, right? And an example I have is, like we're creating glycogen, and glycogen is the storage form of glucose. It's like the most complex form of glucose. So yeah, the absorptive state is associated with GI nutrients being absorbed into the blood and then delivered throughout the body. And then by definition, the absorptive state is associated with the process of anabolism, the synthesis of energy reserves. And an example of that is glycogen, the energy reserve of glucose. So that's the absorptive state. I want someone to tell me now the post-absorptive state. What are the functions associated with that? So A and C are associated with feasting, the absorptive state. What's the post-absorptive state, otherwise known as fasting? What's that associated with? Which functions?
B and D.
Yeah, absolutely. So B and D for post-absorptive. So why is that? Well, post-absorptive, otherwise known as fasting, right? Fasting means we're not eating, we're not consuming anything. So if we think about it conceptually, okay, if the absorptive state, otherwise known as feasting, is we're getting our energy from eating a meal. If we're not eating a meal, right, we're fasting, we still need energy to survive. Like our cells and tissues still need energy to perform its functions. So how are we going to get that energy if it's not coming from food? Well, we can take what we gained from anabolism, right? So in the feasting state, anabolism, the creation of energy reserves, and we're using the example of glycogen, right? We have glycogen stored. If we're not eating, but we need energy, the glycogen is going to be broken down, right? And so B and D are related because they're taking the body's own energy stores, right? Glycogen is the storage form of glucose, and they're going to utilize that. And how specifically is that utilized? Through catabolism, the breakdown of that energy reserve. So in other words, if we use the example of glycogen, we're going to take this storage form of glycogen because we're not eating, but we still need energy. And the glycogen that has already been stored in our bodies is going to break down into glucose. And so having this glucose now in our blood is going to give us that energy. So that's the difference between the two as far as how is energy created, used, and all that. All right, good work. So next slide. All right, so we're going to do a quick overview of the regulation of metabolism, and specifically how glucose is involved. And so so depending on whether glucose levels in the blood specifically are high and low, are going to dictate the actions of two very important hormones. And those hormones are glucagon and insulin. And so overall, these hormones regulate our absorptive and post-absorptive state. So quick anatomy question. Glucagon is a hormone that is secreted by a certain type of cells, certain types of cells in the pancreas. What are those cells called in the pancreas that secrete glucagon?
The alpha cells.
Yes, absolutely. So the alpha cells in the pancreas. So if glucagon is secreted by the alpha cells, insulin is secreted by the which cells in the pancreas?
Beta cells.
Yes, absolutely. So those are the two cells. So alpha is associated, it secretes glucagon, beta secretes insulin, and these are both from the pancreas. And this, the first was alpha and this one is beta. Yes. All right. So glucagon is released when blood glucose is high or low. Can somebody tell me? When blood glucose is high or low, glucagon is released. Which one is it?
Low.
Yes.
Low.
Low. Yes, good job, both of you. So let's think about this conceptually. If blood glucose is low, and therefore glucagon is secreted, right? In which functional state do you think we're in right now? Like which functional state is associated with low blood glucose levels?
Post-absorptive.
Post-absorptive.
Yeah, awesome. So, right, the post-absorptive, we're not consuming a meal, so our blood glucose is on the lower side, right? And so the alpha cells in the pancreas like recognize that and they're like, oh, I'm going to secrete glucagon. And so that's going to be secreted when the blood glucose levels are low. All right, let's focus on insulin. So insulin is released when blood glucose is what? High or low? High. Yes, absolutely. So let's think about this conceptually. If blood glucose is high, what does that tell us about the state that we're in? What does that suggest? Yeah, in the absorptive feasting state, absolutely. So as we're eating a meal, blood glucose is going to spike. And so as blood glucose spikes, that triggers the beta cells of the pancreas to then release insulin. And so to kind of summarize that, we can conclude that the function of glucagon is to raise blood glucose because, right, the blood glucose is low, so glucagon comes in to raise it. And then the function of insulin is to lower blood glucose because it was high. And we'll kind of see how insulin does that when we talk about diabetes. All right, I'm going to clear that. All right, next slide. Or any questions on that before we move on? No? Okay, next slide. So keeping an eye on the time, I'm going to really quickly go over diabetes mellitus. So there are two types of diabetes. There's type 1 diabetes and type 2 diabetes. The difference between the two is that type 1 is an autoimmune disorder. And so what that means is that the immune system like attacks your own body. So for people with type 1 diabetes, their beta cells in their pancreas are actually like non-functionable. They're destroyed by this autoimmune disorder. So let's think about that. If beta cells are non-functioning, they're not going to be secreting insulin, right? Because that's what the beta cells do. They secrete insulin. So if you don't have working beta cells, you're not going to be secreting insulin. And so if you're not secreting insulin, your blood glucose is going to be really high, right? Because what is the one thing, or what is, what lowers blood glucose, right? Insulin, because insulin is a hormone that takes the glucose in the blood and it pushes it into cells. The cells, they want glucose, right? The blood, if there's too much, it's not a good thing, right? The cells will take it, right? And that's where we see this thing of the cellular uptake of glucose. So if glucose is in the blood and insulin is not there to move it into the cells, it's going to stay in the blood and we're going to have high blood sugar, hyperglycemia. So just a summary, beta cells are destroyed, therefore no insulin is there. So that's why type 1 diabetics oftentimes need to obtain insulin therapy so that their body can gain insulin. Type 2 diabetes is a little bit different. In type 2 diabetes, in type 2 diabetics, the beta cells are okay. They're not autoimmunely destroyed or anything. So type 2 diabetics, their body makes insulin. So insulin is there, but the cells that need to respond to the insulin, the cells that need to take that blood glucose and put it into the cells, they're like resistant. They don't recognize that, all right, insulin's here and we need to uptake the glucose. The cells don't respond. And so if the cells aren't responding and the cells aren't taking in the glucose, where's the glucose going to go? It's going to stay in the blood. And so therefore, if you have high glucose in the blood, hyperglycemia, and that's also diabetes. So that's the difference between type 1 and type 2, just a quick summary. So we'll go to the next slide.
Okay, so reproductive system. So we're running a little bit short on time, but that's no worries but I still want to have allow 5 minutes for discussion or questions. So for reproductive system, I'm going to go over just some key terminologies and reproductive endocrinology. Okay. So some common key terms. So let's first start with the gonads. So the gonads are our primary reproductive organs, right? So for the males, the gonads are the testes. For females, the gonads are the ovaries. And these gonads are responsible for producing sperm and egg, like sperm for male and egg or ovum for females. And these gonads can also secrete hormones, okay? Next, we have gametes. Gametes is a mature male or female germ cells, right? They're sperm and egg, right? So when once a male and a female gamete unite during sexual reproduction, a zygote can be formed. Okay. All right. And then we have gametogenesis. So whenever you see a term that ends in genesis, just know that it often refers to the production or the formation of something. So gametogenesis literally means the production of gametes. Okay. And the two types of gametogenesis in this chapter would be spermatogenesis and oogenesis. Okay. And then there are also gonadotropic hormones. If you remember in your endocrine glands chapter, I don't know if it's maybe chapter 11, right? So whenever a hormone name ends in tropic, it's tropic hormones are hormones that control the blood concentration of another hormone, right? So gonadotropic hormones, right, which are the follicle stimulating hormone and the luteinizing hormone, they're going to control the secretion of another hormones. All right. Then we have gonadosteroids. These are sex steroid hormones, right? They're testosterone, estradiol, and progesterone. They stimulate reproductive organs and germ cell maturations and other functions in both males and females. So these are just some common terminology, okay? So I'm not going to cover sexual reproduction, so I'm going to skip to reproductive endocrinology, okay? So during puberty, the gonads secrete more sex steroids hormones due to stimulation by follicle stimulating hormone, and luteinizing hormone from the anterior pituitary gland. Okay. Remember the sex steroid hormones are testosterone, estradiol, and progesterone, right? And they're stimulated by these gonadotropic hormones, right? The follicle stimulating hormone and luteinizing hormone from the anterior pituitary gland. And it involves a 3 hormone sequence, right? Remember from chapter 11, endocrine glands, we talked about the 3 steps of the 3 hormone sequence, right? Where it starts in the hypothalamus and then to the anterior pituitary gland. And then those hormones transfer to our target cells where they stimulate or inhibit certain action. Okay. So first, in the reproductive system, the hypothalamus secretes a hypothalamic hormone, right? Which is gonadotropin releasing hormone in this case. All right. Now, this gonadotropin releasing hormone will then stimulate the anterior pituitary gland to secrete more hormones, to secrete the follicle stimulating hormone and luteinizing hormone. All right. And these gonadotropic hormones, right, follicle stimulating hormone and luteinizing hormone are considered gonadotropic hormones. Now, these hormones are going to stimulate the gonads. They're going to travel to the gonads and stimulate them to secrete sex hormones. Okay. So they're going to secrete sex hormones, which are testosterone, estradiol, and progesterone. Right? We talked about this. And they're also going to undergo gametogenesis, right, which is the formation or the production or formation of new sperm or new eggs, right, depending on its sex. And there are a few practice questions I want you guys to go over. And these are like the type of questions that will be on the exam, which is why I want you guys to have a chance to get a feel of what kind of questions that Dr. Arnaud would ask. Okay, so in the reproductive system, the hypothalamus secretes what hormone, which stimulates the anterior pituitary gland to secrete what hormone? And read the questions carefully, keep in mind of the 3 hormone sequence, right? The hypothalamus secretes a hypothalamic hormone, and the anterior pituitary gland secretes an anterior pituitary hormone, right? You guys about 10 more seconds. Okay, I'm gonna end the poll. So majority of you said it's B, and majority of you said it's A. The correct answer to this is B. All right, remember, keep in mind the 3 hormone sequence, right? The hypothalamus secretes the gonadotropin releasing hormone, which this hormone would stimulate the anterior pituitary gland to secrete follicle stimulating hormone. So keep in mind, like these gonadotropic hormone, these follicle stimulating hormone and luteinizing hormone are secreted only by the anterior pituitary gland, not the hypothalamus. Okay, the hypothalamus only secretes the gonadotropin releasing hormone, which stimulates the anterior pituitary gland to secrete follicle stimulating hormone and luteinizing hormone. Okay, you should understand the 3 hormone sequence for the reproductive system. One more question. This is the last question, all right? So gametogenesis is directly stimulated by which hormone? And remember, gametogenesis is the production of gametes, right? And gametogenesis is stimulated by what hormone? We've talked about this. And a hint I will give you is that this hormone is often referred to the gonadotropic hormone. Give you guys about 10, 15 more seconds. Okay, I'm gonna end the poll now. So the correct answer to this question is, oops, D. The correct answer is D, follicle stimulating hormone. Remember, if I can go back to the previous slides. So here's the 3 hormone sequence, right? And we talked about gametogenesis. Gametogenesis occurred in the gonads, right? And the gonads are stimulated, right? Our gonads are stimulated by the follicle stimulating hormone and luteinizing hormone, right? So I would suggest you to differentiate between what types of hormone is secreted by the hypothalamus and what hormones is secreted by the anterior pituitary gland, and what are their functions, right? What do they stimulate and what do they secrete? Okay, so keep in mind that, and that should end our workshop presentation. Do you have anything to add, Stacy?
No, that was great. You just, if you guys have any questions, we'll be on for a few minutes.
Yeah, so there are 3 more minutes left for our workshop. So you guys want to stay, feel free to stay to ask questions or discuss about the materials we've covered, or feel free to hop off.
Good luck on finals, everyone.
And keep in mind, please, complete the surveys.
Okay.
Complete the survey that is posted on the chat right now. And once you complete the survey, we will email you the slides with the answers, of course. And you guys can refer back to the workshop materials if you need.
On finals, everybody.
Good luck with finals.
The recording will be on YouTube, correct?
Yes.
Yeah, I put the link to the channel in the chat. So that's the college's YouTube channel. So it has, every time we do a workshop, it goes on there. So this one, as well as like Bio 65, Stats, Chem. So like all our workshops for finals were today. So those will be uploaded either today or sometime this week. It should be soon. But then there's also like our mid-semester ones. And like other finals workshops from previous semesters, which cover similar topics and chapters. They're all on that YouTube link.
Okay, thank you.
Thank you.
Good luck on finals.
Thank you. You too.
Let's stop the share or stop the recording.